Welcome to MedHub. Today, we're diving into the fascinating world of human DNA, the blueprint of life that makes each of us unique. From its intricate double helix structure to the groundbreaking discoveries shaping our understanding of genetics, this video will unravel the mysteries of DNA like never before. Let's get started. Deoxyribonucleic acid is a polymer composed of two polynucleotide chains that coil around each other to form a double helix. The polymer carries genetic instructions for the development, functioning, growth, and reproduction of all known organisms and many viruses. DNA and ribonucleic acid, RNA, are nucleic acids. Alongside proteins, lipids, and complex carbohydrates, polysaccharides, nucleic acids are one of the four major types of macromolecules that are essential for all known forms of life. Properties of human DNA structure. Human DNA, deoxyribonucleic acid, is a double helical molecule that carries the genetic instructions essential for the development, function, growth, and reproduction of humans. We are going to discuss the detailed properties, with a focus on structural and functional aspects specific to humans. 1. Double helix structure. Human DNA shares the iconic double helix structure described by Watson and Crick. The two strands coil around a central axis, forming a right-handed helix. It's stabilized by hydrogen bonds between complementary bases, AT and GC, and hydrophobic interactions between stacked bases. Human DNA composed of nitrogenous bases, purines which are adenine, A, and guanine, G, and pyrimidines which are thymine, T, and cytosine, C. They are paired as adenine pairs with thymine via two hydrogen bonds. Guanine pairs with cytosine via 3 hydrogen bonds. Human DNA is approximately 40% GC and 60% AT, though this varies by region. The two strands have anti-parallel orientation. They run opposite directions. One strand runs 5th to 3rd, and the other runs 3rd to 5th. This polarity is crucial for replication and enzymatic activities like transcription. Sugar phosphate backbone. Composed of sugar deoxyribose phosphate groups link adjacent sugars via phosphodiester bonds. It provides structural integrity and protection to the DNA molecule. Chromosomal organization. The complete set of DNA in humans is known as the human genome. Human DNA is packaged into 23 pairs of chromosomes, 46 in total. Consist of autosomes, which are 22 pairs, and sex chromosomes, which is 1 pair, XX in females, XY in males. Each chromosome contains a single, long DNA molecule tightly coiled around histone proteins. Nucleosomes are further folded to create chromatin, which condenses to form chromosomes during cell division. The human genome consists of 3 billion base pairs. If stretched out, the DNA in a single human cell measures approximately 2 meters in length. DNA exhibits structural flexibility to accommodate cellular processes. It has supercoiling ability, in which DNA is twisted into compact forms for efficient storage, and relaxed states which occur during replication or transcription. DNA is unwound by enzymes like helicases. During replication, each daughter DNA contains one parent strand and one newly synthesized. Also, human DNA has multiple origins of replication to facilitate rapid duplication of its large genome. Human DNA has lot of genetic variabilities. Single nucleotide polymorphisms. SNPs. Variations in a single base that contribute to genetic diversity. Copy number variations. CNVs. Variations in the number of copies of certain DNA segments. Mutations. Changes in DNA. Sequences that can lead to genetic disorders or evolution. Major and minor grooves. The double helix has major groove, which is wider and accessible for binding by transcription factors, and minor groove, which is narrower and less accessible. These grooves are crucial for protein-DNA interactions. Charged nature. The phosphate groups in the backbone give DNA a negative charge. This property allows DNA to interact with positively charged histones and facilitates separation in techniques like gel electrophoresis. Stability. Guanine cytosine content, higher GC content in certain regions increases DNA stability due to stronger hydrogen bonding. Packaging. DNA is compacted into nucleosomes and higher-order structures to protect it from damage. A gene is the basic functional unit of heredity, composed of DNA sequences that encode the instructions for synthesizing RNA and proteins. The human genome, housed in the cell's nucleus and mitochondria, contains approximately 20,000 to 25,000 protein-coding genes and a vast number of non-coding genes that play crucial regulatory roles. Structure of a gene. Human genes are organized into several key regions. Coding regions. Exons. These are sequences that code for amino acids and are transcribed into mRNA. They are spliced together to form the final transcript. Non-coding regions. Introns. Non-coding sequences within a gene that are removed during RNA processing. Regulatory sequences. Promoters. Located upstream of the transcription start site. Promoters contain binding sites for RNA polymerase and transcription factors. Enhancers. Silencers. Regulate the gene's expression from a distance, often through chromatin looping. Untranslated regions, UTRs, found at both the fifth and third ends of mRNA. UTRs play roles in translation, regulation, mRNA stability, and localization. Gene function. Human genes serve a variety of roles, including protein synthesis. Genes encode proteins that perform structural, enzymatic, signaling, and transport functions. Regulation of cellular processes. Genes regulate growth, development, metabolism, and homeostasis. For example, tumor suppressor genes, TP53, regulate cell division and prevent cancer. Response to environmental stimuli. Genes encode proteins and RNAs that help cells respond to external changes, such as heat shock proteins under stress. Hereditary transmission. Genes carry the information passed from parents to offspring. Types of DNA. Human DNA can be classified into different types based on its location, structure, function, and sequence characteristics. The main types of DNA in humans include nuclear DNA, mitochondrial DNA, and variations based on coding, non-coding, and repetitive regions and structure. Here is detailed breakdown based on location. 1. Nuclear DNA, found in the nucleus of eukaryotic cells, accounts for nearly 99.9% of the total DNA in humans, organized into 46 chromosomes, 23 pairs, 22 pairs of autosomes, 1 pair of sex chromosomes, XX in females, XY in males. encodes the majority of the genetic information required for cellular function and development. Features of nuclear DNA. It's double-stranded, linear DNA molecules, diploid, having 2 copies of each chromosome, 1 from each parent. Genetic content are protein-coding genes, 20,000 to 25,000 genes. Regulatory elements are promoters, enhancers. Non-coding regions are introns, pseudogenes. Function governs the synthesis of proteins via transcription and translation. Regulates cell cycle, growth, and reproduction. 2. Mitochondrial DNA, mtDNA. Found in the mitochondria, the energy-producing organelles of the cell. Accounts for 0.1% of the total DNA in humans. Maternally inherited, transmitted exclusively from the mother. Features of mtDNA. 1. Circular structure, double-stranded, circular DNA. 2. Small size, contains 16,569 base pairs and 37 genes. 3. Genes. 13 genes encode proteins for oxidative phosphorylation, ATP, production. 22 tRNA genes and 2 rRNA genes for mitochondrial protein synthesis. 4. Function. Essential for cellular energy metabolism. 5. High mutation rate. mtDNA mutates more frequently than nuclear DNA, making it useful in evolutionary and forensic studies. Based on coding potential. Coding DNA makes up 1.5% of the human genome. Contains exons, which are sequences directly transcribed and translated into proteins. Features of coding DNA. 1. Encodes proteins that perform structural, enzymatic, and regulatory functions. 2. Includes genes for enzymes, hormones, receptors, and more. Non-coding DNA comprises 98.5% of the human genome. Does not code for proteins but plays critical regulatory and structural roles. Types of non-coding DNA. 1. Regulatory DNA includes promoters, enhancers, silencers, and insulators. Controls gene expression and transcription. 2. Introns, non-coding sequences within genes, removed during RNA splicing. 3. Pseudogenes, non-functional copies of genes, often remnants of evolutionary changes. 4. Functional. Non-coding RNA, ncRNA, includes tRNA, rRNA, microRNA, and lncRNA. Involved in protein synthesis, regulation of gene expression, and chromatin structure. Based on sequence characteristics. Accounts for 50% of the human genome. Consists of repeated sequences, with structural, regulatory, or unknown functions. 1. Tandem repeats. Short sequences repeated adjacently. Includes microsatellites, short tandem repeats, STRs, and mini-satellites. A microsatellite is a short tandem repeat with a length of 1 to 6 base pairs or more, which is frequently repeated 5 to 50 times. A mini-satellite is similar to a tiny tandem repeat in that it is repeated 5 to 50 times, but with considerably longer base pairs, from 10 to 60. Used in forensic DNA fingerprinting. 2. Interspersed repeats. Scattered throughout the genome. Includes transposable elements like lines, long interspersed nuclear elements, and signs, short interspersed nuclear elements. 3. Satellite DNA. Found in centromeres and telomeres, contributing to chromosomal stability. Based on modifications. DNA can undergo chemical modifications that alter its function without changing the sequence. Types of modifications. 1. Methylated DNA. DNA methylation, addition of a methyl group, at CpG islands, regulates gene expression. Here is the process of DNA methylation. DNA methylation usually happens in CpG islands by adding a methyl group, provided by S-adenosylmethionine, SAM, to the carbon-5 position of a cytosine. This process is catalyzed by DNA methyltransferases, DNMTs. DNA methylation of gene promoters. Unmethylated CpGs result in gene activation. Methylated CpGs lead to gene silencing. 2. Hydroxymethylated DNA. Involved in active DNA demethylation and gene regulation. Based on the structure. Human DNA exists in various structural forms depending on the environmental conditions and biological context. These structural types primarily refer to the three-dimensional conformations of the DNA molecule. The structure can influence its function, replication, and interaction with proteins. Below are the key structural types of human DNA. 1. bDNA, canonical form. It is the most common form, predominantly found under physiological conditions, normal temperature and salt concentrations. It is right-handed helix, the strands twist around the axis in a right-handed manner. Its dimensions are: helical rise per base pair is 3.4 angstroms, 10.5 base pairs per turn, and diameter 2 nanometers. Its major groove is wide and deep, facilitating protein binding, and minor groove is narrow and shallow. Biological role. The standard form of DNA involved in replication, transcription, and interaction with regulatory proteins. 2. aDNA. Features: right-handed helix, similar to bDNA but more compact. Environment: found under dehydrated conditions or in DNA-RNA hybrids and RNA duplexes. Dimensions: helical rise per base pair 2.3 angstrom, 11 base pairs per turn. Diameter 2.6 nanometers, wider than bDNA. Grooves: major groove, deep and narrow. Minor groove, broad and shallow. Biological role: commonly seen in crystallized DNA samples and during interactions involving DNA and RNA. 3. zDNA. Its features are: left-handed helix. The strands twist in a left-handed manner, opposite to bDNA and aDNA. Environment: forms in regions rich in GC repeats or under high salt concentrations and torsional stress. Dimensions: helical rise per base pair 3.7 angstrom, 12 base pairs per turn. Diameter 1.8 nanometers, narrower than bDNA. No distinct major or minor grooves. Biological role: plays a role in gene regulation and chromatin remodeling. Acts as a marker for specific protein-DNA interactions and participates in stress response. 4. Single-stranded DNA, ssDNA. Features: occurs transiently during replication, transcription, recombination, and repair. Lacks a double helix structure. Exists as a flexible, linear strand. Stabilized by proteins like single-strand binding proteins, SSBs, during replication. Biological role: intermediate in DNA replication. Template for complementary strand synthesis. 5. Triplex DNA. hDNA. Features: triple-stranded structure, formed when a third strand binds to the major groove of duplex DNA via Hoogsteen hydrogen bonds. Common in regions with purine-rich or pyrimidine-rich sequences. Biological role: may regulate transcription, involved in recombination and DNA repair processes. 6. Quadruplex DNA. G quadruplexes. Its features are: four-stranded structure, formed by guanine-rich sequences. G-tracts. Stabilized by Hoogsteen hydrogen bonding, forming G-tetrads. Found in specific genomic regions like telomeres. Promoters of oncogenes, for example, CMYC, KRAS. Biological role: regulates transcription and telomere maintenance. A potential target for anti-cancer therapies. 7. Circular DNA. Its features are: found in mitochondrial DNA and extrachromosomal. Circular DNA lacks free ends, reducing vulnerability to exonucleases. Can form supercoiled structures to fit within organelles. Biological role: mitochondrial energy production. Structural support in some cancer cells with amplified oncogenes. 8. Hairpin and cruciform DNA. Its features are: formed in regions with palindromic sequences. Hairpin DNA. A single strand folds back on itself to form a stem loop structure. Cruciform DNA. Double-stranded DNA forms cross-shaped structures. Biological role: seen in regulatory regions. May play a role in DNA replication and recombination. 9. Non-BDNA structures. Regions of the genome can adopt non-B forms, which include slipped strand DNA, found in repeat regions during replication. Slippage. R loops, formed during transcription when the RNA strand hybridizes with the DNA. Template strand. 10. Hybrid DNA. Its features are: it formed during DNA replication or transcription when RNA temporarily pairs with DNA, DNA-RNA hybrids. Found in Okazaki fragments. On the lagging strand. Regions with active transcription. Biological role: it stabilizes replication intermediates and facilitates transcription termination in certain contexts. 11. Supercoiled DNA. Features: highly compacted DNA seen in both nuclear and mitochondrial DNA. Positive supercoiling. Overwinding of the DNA helix. Negative supercoiling. Underwinding. Promoting strand separation. Managed by enzymes like topoisomerases. Biological role: facilitates DNA packaging and access during transcription and replication. Based on function: telomeric DNA. Specialized DNA found at the ends of chromosomes. Composed of repetitive sequences, TTAGGG in humans. Protects chromosomes from degradation and prevents fusion with other chromosomes. Centromeric DNA. Found at the centromere, the constricted region of chromosomes. Contains repetitive sequences and is crucial for spindle attachment during cell division. Extrachromosomal DNA. Small DNA molecules not part of chromosomal DNA. Includes circular mtDNA in mitochondria. Extrachromosomal DNA, ecDNA, in cancer cells, which can amplify oncogenes. Functions of human DNA. Human DNA is the blueprint of life, storing, transmitting, and regulating the genetic information required for the development, functioning, and reproduction of living organisms. Its functions can be broadly categorized into genetic, structural, and regulatory roles. Storage of genetic information. DNA is the repository of all genetic information required to build and maintain an organism. Encodes instructions for synthesizing all proteins, which perform vital functions in the body. Stores hereditary information in the sequence of its nucleotide bases, adenine, thymine, guanine, and cytosine. Genetic information is organized into genes, with each gene coding for a specific protein or functional RNA. Replication. DNA serves as a template for its own duplication, ensuring that genetic information is passed to the next generation of cells. Replication occurs during the S phase of the cell cycle. This process ensures continuity of genetic information during cell division, accurate transmission of genetic material from parent to daughter cells, transcription and translation, gene expression. DNA directs the synthesis of RNA and proteins, which are essential for cellular functions. Transcription. DNA is transcribed into messenger RNA, mRNA, in the nucleus. mRNA carries genetic instructions to the ribosome. Translation. mRNA is translated into a specific protein sequence at the ribosome. Proteins perform structural, enzymatic, and regulatory functions in the body. Regulation of gene expression. DNA contains regulatory sequences that control the timing, location, and amount of gene expression. Promoters and enhancers facilitate or inhibit the binding of transcription factors. Epigenetic modifications. DNA methylation silences genes. Histone acetylation activates genes, allows cells to respond to environmental changes and signals, for example hormones, stress. Cellular differentiation. DNA ensures that specialized cells, for example, neurons, muscle cells, express only the genes relevant to their function. Regulation of gene expression allows multicellular organisms to develop distinct tissues and organs. Maintenance of genome stability. DNA performs functions that safeguard the genome's integrity. DNA repair fixes damage caused by replication errors, UV light, chemicals, or radiation. Mechanisms include mismatch repair, base excision repair, and nucleotide excision repair. Telomeres protect the ends of chromosomes from degradation and fusion with other chromosomes. Heredity and evolution. DNA ensures genetic continuity across generations and contributes to evolutionary changes. Inheritance. Nuclear DNA is inherited from both parents, while mitochondrial DNA is maternally inherited. Mutations. Changes in DNA sequences lead to genetic variation. Beneficial mutations drive evolution and adaptation. Structural function. DNA contributes to the structure of chromosomes and chromatin. Chromatin. DNA is wrapped around histone proteins to form chromatin, allowing efficient packaging in the nucleus. Chromosomes serve as the physical carriers of genetic material during cell division. Maintain structural integrity of the genome. Role in cellular processes. DNA participates in various cellular processes beyond replication and transcription. Cell cycle. Regulation. DNA controls the checkpoints ensuring proper cell division. Apoptosis. Damaged or irreparable DNA can trigger programmed cell death to prevent the proliferation of faulty cells. Production of functional RNAs. DNA encodes various functional RNA molecules that play crucial roles in cellular functions. rRNA, ribosomal RNA, forms the core of ribosomes. tRNA, transfer RNA, brings amino acids to ribosomes during translation. mRNA, microRNA, and siRNA, small interfering RNA, regulate gene expression by targeting mRNAs for degradation or inhibition. LNcRNA, long non-coding RNA, involved in gene regulation, chromatin remodeling, and epigenetics. DNA in medicine and biotechnology. Human DNA serves as the foundation for various medical and technological advancements. Forensics. DNA profiling identifies individuals based on their unique genetic patterns. Gene therapy corrects genetic disorders by inserting functional genes into cells. Biotechnology. DNA is used in cloning, CRISPR gene editing, and synthetic biology. Role in adaptation and stress response. DNA helps organisms adapt to environmental stress. Heat shock genes, activated under stress to protect cellular proteins. Stress-induced mutations can lead to genetic diversity and survival in challenging environments. DNA replication. In molecular biology, DNA replication is the biological process of producing two identical replicas of DNA from one original DNA molecule. DNA replication occurs in all living organisms, acting as the most essential part of biological inheritance. This is essential for cell division during growth and repair of damaged tissues, while it also ensures that each of the new cells receives its own copy of the DNA. The cell possesses the distinctive property of division, which makes replication of DNA essential. Characteristics of human DNA. Replication. Semi-conservative replication. Each new DNA molecule consists of one original parental strand and one newly synthesized strand. Bidirectional replication. Replication proceeds in both directions from multiple origins of replication to speed up the process. Multiple origins of replication. Human DNA has multiple origins of replication due to its large genome, 3 billion base pairs, ensuring timely replication. Template-driven. Each strand of the DNA double helix serves as a template for the synthesis of a complementary strand. Strict regulation. Replication is tightly regulated by the cell cycle to prevent errors and ensure proper duplication. 2 stages of DNA replication in humans. A. Initiation. First, to know the origin of replication. A. Sequence. Features. Have multiple origins of replication per chromosome to replicate large genomes efficiently. Origin sequences are less conserved than in prokaryotes. Frequently associated with at-rich regions for easier unwinding. Replication timing zones, early or late. Replication. B. Replication bubble formation. At the origin, DNA strands separate to form a replication bubble with two replication forks proceeding in opposite directions. See bidirectional replication. Replication is generally bidirectional from the origin, ensuring rapid and complete genome duplication. 2. Licensing the origin. A. Identification of origins. Origins of replication are specific DNA sequences where replication begins. These regions are recognized by the Origin Recognition Complex, ORC, a 6-subunit protein complex, ORC1 ORC6. ORC binds to the origin throughout the cell cycle but is active only during the G1 phase. B. Recruitment of licensing factors. Its steps include 1. Formation of pre-recognition complex. The ORC recruits additional proteins to form the pre-RC. During the G1 phase, the pre-RC is assembled at origins of replication. Key factors include CDC6, cell division cycle 6, binds to ORC and helps recruit MCM helicase. CDT1, chromatin licensing and DNA replication factor 1, loads the MCM helicase onto DNA. 2. Loading of MCM helicase. The minichromosome maintenance, MCM, complex, composed of MCM2 MCM7 subunits, is a key helicase responsible for unwinding DNA during replication. MCM is loaded onto the DNA as an inactive double hexamer. 3. Regulation of origin licensing. A. Prevention of relicensing. 1. Cell cycle dependent inhibition. Origin licensing is restricted to the G1 phase. High CDK activity in S, G2, and M phases prevents relicensing by inhibiting key licensing factors like CDC6 and CDT1. 2. Proteolysis. CDT1 is degraded during the S phase to prevent relicensing. The E3 ubiquitin ligase SCF, SKP2, targets CDT1 for degradation. 3. Geminin inhibition. Geminin, a regulatory protein, binds to CDT1 during the S phase, blocking its interaction with the ORC. B. Ensuring fidelity. Only properly licensed origins can initiate replication. Licensing defects can lead to genomic instability, replication stress, or incomplete DNA duplication. 3. Activation of replication origins. As we know about the formation of pre-replication complex and its key components, the pre-recognition complex marks the origin for potential activation during the S phase but remains inactive until specific signals are received. A. Helicase activation. The key event in origin activation is the conversion of the inactive MCM helicase into an active helicase capable of unwinding DNA. This involves 1. Phosphorylation by DDK, CDC7-DBF4. DDK phosphorylates the MCM complex, priming it for activation. This phosphorylation destabilizes inhibitory interactions within the MCM complex. 2. Phosphorylation by CDKs recruits replication factors like CDC45 and GINs to form the active helicase complex, CMG complex, CDC45 MCM GINs. B. Recruitment of replication factors. Once phosphorylated, the MCM helicase recruits additional factors to form the active CMG complex. CDC45, essential for helicase activation and recruitment of DNA polymerases. GINs complex, SLD5, PSF1, PSF2, PSF3, stabilizes the MCM helicase and aids in fork progression. The CMG complex, CDC45, MCM, GINs, is the fully active helicase that unwinds the DNA at the origin, creating single-stranded DNA templates for replication. 4. Single-strand stabilization. DNA. Helicase unwinds the double-stranded DNA, dsDNA, at the replication fork, creating single-stranded DNA, ssDNA. The exposed ssDNA is prone to re-annealing with its complementary strand. Formation of secondary structures, for example hairpins or loops. Degradation by nucleases. Stabilization of ssDNA ensures efficient replication by keeping the strands accessible. For DNA polymerases, helicase unwinds the double-strand DNA, exposing ssDNA at the replication fork. Replication protein or immediately binds to the exposed ssDNA. The replication protein A is the key in ssDNA stabilization. RPA prevents the formation of hairpins, loops, or other structures that could block replication machinery. RPA shields the single-strand DNA from reactive molecules and nucleases. RPA interacts with other proteins, including DNA polymerase alpha primase complex, to initiate replication. Proliferating cell nuclear antigen (pCNA) to coordinate polymerase activity. Replication fork stabilizers to maintain fork integrity. Elongation. 1. Primer synthesis. Primer synthesis is a critical step during DNA replication in humans, where short RNA primers are synthesized to provide a starting point for DNA polymerases. Since DNA polymerases cannot initiate synthesis de novo, primers are essential for replication. Initiation. Steps in primer synthesis are Step 1. Helicase unwinding. The MCM complex, minichromosome maintenance helicase, unwinds the double-stranded DNA, creating a replication fork with single-stranded DNA, ssDNA, templates. Step 2. Binding of the primase. Primase, Prim1 and Prim2, recognizes the ssDNA at the replication fork and binds to the template strand. Step 3. RNA. Primer synthesis. Primase synthesizes a short RNA primer, about 8 to 12 nucleotides, complementary to the DNA template strand. RNA primers are laid down in both leading and lagging strands. Leading strand. One RNA primer is synthesized at the origin of replication. Lagging strand. Multiple RNA primers are synthesized periodically for Okazaki fragment initiation. Step 4. Primer extension by DNA polymerase alpha. The catalytic subunit of DNA polymerase alpha, polar 1, extends the RNA primer with a short stretch of DNA, about 20 nucleotides. This RNA-DNA hybrid primer serves as the substrate for high-fidelity DNA polymerases, for example, polymerase delta and sigma. 4. Role of primers in leading and lagging strand synthesis. A. Leading strand requires a single primer at the origin of replication to initiate continuous synthesis. B. Lagging strand requires multiple primers due to the discontinuous nature of synthesis. Each Okazaki fragment, 100 to 200 nucleotides in humans, begins with an rDNA primer. Regulation of primer synthesis. A. Temporal regulation. Primer synthesis is tightly coordinated with helicase activity and replication fork progression. B. Coordination with other proteins. Replication protein A. RPA stabilizes ssDNA and prevents secondary structure formation until the primase binds. pCNA. Proliferating cell nuclear antigen. Recruited after primer extension to coordinate polymerase switching. 2. DNA synthesis. Leading strand. DNA polymerase epsilon synthesizes the leading strand continuously in the fifth to third direction, following the replication fork. Lagging strand. DNA polymerase delta synthesizes the lagging strand discontinuously as short Okazaki fragments, about 100 to 200 nucleotides in humans. 3. Clamp loading. Step 1 is called clamp loader activation. In this step, the replication factor C is a multi-subunit protein complex responsible for opening and loading the sliding clamp onto DNA. It binds to ATP molecules, undergoing a conformational change that prepares it to interact with pCNA. Next, step 2 is pCNA binding. RFC binds to pCNA, causing the ring structure of pCNA to open at one of its subunit interfaces. Step 3 is DNA recognition. The RFC pCNA complex recognizes and binds to the primer template junction on the DNA. The primer template junction is a site where single-stranded DNA meets double-stranded DNA. Step 4 is pCNA loading. The open pCNA ring is placed around the DNA strand at the primer template junction. Once positioned, pCNA closes around the DNA, encircling it. Step 5 is ATP hydrolysis. RFC hydrolyzes ATP, leading to its dissociation from pCNA and the DNA strand. This step locks pCNA onto the DNA and releases the RFC clamp loader. Step is DNA polymerase recruitment. DNA polymerases delta and sigma recognize and bind to pCNA, initiating or resuming DNA synthesis. Coordination of leading and lagging strand synthesis. Leading strand pCNA is loaded once at the origin of replication and remains associated with the DNA polymerase epsilon throughout continuous synthesis. Lagging strand pCNA is repeatedly loaded onto the DNA for each Okazaki fragment. As DNA polymerase delta synthesizes the fragments discontinuously. 4. Topological stress relief. Nature of topological stress. A. Supercoiling. B. DNA entanglement. C. Replication fork. Stalling. Steps in topological stress relief. Fig E shows unwinding of the duplex DNA generates a positive supercoil ahead of the replication fork, RF, which is removed mainly by topoisomerase 1, top 1, but also by top 2 or precatenanes behind the RF, the decatenation of which requires top 2. Figure B shows that positive supercoils must be converted into precatenanes at replication termination. As the RFs approach one another before fusion, the region available to contain positive supercoils diminishes and precatenanes form. Figure C show the head-on encounter of a RF with a transcription unit leads to RF demise. The predominant topoisomerase involved in each situation is shown in red and the other in yellow. 4. Coordination with other replication machinery. Topological stress relief is integrated with the activity of other proteins in the replication machinery. Helicase, MCM complex, generates unwinding stress that topoisomerases alleviate. Sliding clamp, pCNA, recruits topoisomerases to specific sites of replication. Stress. Replication fork proteins stabilize the fork and ensure that topoisomerases function efficiently. 5. Primer removal. In DNA replication, primers are short RNA sequences synthesized by primase, a component of DNA polymerase alpha, to provide a starting point for DNA polymerase to initiate DNA synthesis. After elongation, these RNA primers must be removed and replaced with DNA to ensure the continuity and integrity of the newly synthesized strands. Primer removal is a highly coordinated process involving several specialized enzymes and mechanisms. In primer removal process in lagging strand, The DNA polymerase delta displaces the RNA primer as it extends the upstream Okazaki fragment, forming a single-stranded 5' RNA or RNA-DNA flap. RNase H2 cleaves the RNA at the rDNA junction, leaving a small flap. Fen1 recognizes and removes the remaining flap, ensuring complete primer excision. DNA polymerase delta synthesizes DNA to fill the gap left by primer removal. DNA ligase I seals the nick, creating a continuous DNA strand. 4. Leading strand. Since only one primer is required to initiate replication, the same mechanism is used to remove the RNA primer and replace it with DNA at the origin of replication. Challenges and errors in primer removal. Incomplete primer removal. Retention of RNA fragments can lead to genomic instability or mutations. Errors in primer removal are implicated in certain cancers and other genetic disorders. Mutations in enzymes. Mutations in RNase H2 or FEN1 can result in defective primer removal, causing replication stress. For example, defects in RNase H2 are linked to Aicardi-Goutieres syndrome, a rare auto-inflammatory disorder. Primer removal and Okazaki fragment. Maturation. Primer removal is integral to the maturation of Okazaki fragments. The process ensures accurate processing of short DNA fragments, elimination of RNA residues, replaced seamlessly by DNA, ligature of fragments into a continuous DNA strand. 6. Ligation is the process of sealing nicks or gaps in the newly synthesized DNA strands to form a continuous, double-stranded DNA molecule. In humans, DNA ligase I is the primary enzyme responsible for this step during DNA replication, particularly on the lagging strand, where multiple nicks are created due to the discontinuous synthesis of Okazaki fragments. Role of ligation in DNA replication. A. Leading strand. DNA ligation is minimal on the leading strand because replication is continuous. A single nick is sealed at the origin of replication after the RNA primer is removed and replaced with DNA. B. Lagging strand. DNA ligation plays a major role in sealing nicks between adjacent Okazaki fragments. Each Okazaki fragment, approximately 100 to 200 nucleotides in humans, is synthesized discontinuously, leaving multiple nicks that must be joined. Mechanism of ligation. Ligation occurs in three main steps. DNA ligase binds to the nick in the DNA strand. The nick has a free third hydroxyl group, OH, and a fifth phosphate group, P, on adjacent nucleotides. Step 1 is adenylation, in which DNA ligase is adenylated by ATP, forming a covalent intermediate with an AMP group. Step 2 is the transfer of AMP. ATP is hydrolyzed to AMP and pyrophosphate, PPI. The AMP group is transferred to the 5' phosphate of the nicked DNA. Step 3 is phosphodiester bond formation, in which the third hydroxyl group attacks the phosphate, forming a phosphodiester bond and releasing AMP. This seals the nick and restores the integrity of the DNA backbone. Last step in DNA replication is termination. Termination of DNA replication is the final stage of the replication process, where the replication machinery completes the synthesis of DNA, resolves any remaining issues, and disassembles. Unlike prokaryotic systems, where termination occurs at specific tersites, termination in human DNA replication is more dynamic and influenced by various factors, including the completion of replication, fork convergence, and telomere replication. First step is the termination at the replication fork. Termination occurs when two replication forks meet. During bidirectional replication, the leading strand of one fork meets the lagging strand of the other fork, completing DNA synthesis. The convergence of forks can generate supercoils and catenanes, interlinked DNA strands. Topoisomerase II alpha resolves these structures by introducing double strand breaks, allowing one DNA duplex to pass through the other and separating the daughter strands. After DNA synthesis is complete, the replication machinery, including the CMG helicase, CDC45, MCM complex, and GINs, is disassembled by the ubiquitin proteasome system. Now, termination at telomeres. Telomeres, the ends of linear chromosomes, are composed of repetitive sequences, TTAGGG in humans, and require specialized handling to ensure complete replication. The lagging strand cannot be fully synthesized at the telomeres due to the inability of DNA polymerase to fill in the final gap left after primer removal. Telomerase, a ribonucleoprotein enzyme, extends the third end of the telomere by adding repetitive sequences using its RNA template. This ensures the lagging strand can be fully replicated without loss of genetic information. After replication, telomeres are capped by the shelterin complex, which protects them from being recognized as DNA breaks and prevents chromosomal instability. That's the end of DNA replication process. Disorders related to DNA. Genetic disorders. Examples include cystic fibrosis, sickle cell anemia, and Huntington's disease. Chromosomal abnormalities. Disorders such as Down syndrome, trisomy 21, and Turner syndrome arise from chromosomal changes. Cancer. Mutations in DNA can lead to uncontrolled cell division, causing cancer. DNA variation. DNA variation refers to differences in the DNA sequence among individuals within a population or species. These variations contribute to genetic diversity, influencing physical traits, susceptibility to diseases, and responses to environmental factors. Variation may be due to polymorphisms, which are small variations in DNA sequences, such as single nucleotide polymorphisms, SNPs, contribute to individual differences, and genetic diversity. Mutations. Changes in DNA sequence can result in genetic disorders or contribute to evolution. Mutations can be inherited or acquired, for example due to environmental factors. Thank you for joining me on this journey into the fascinating world of human DNA. We've explored its structure, functions, and unique features, uncovering how this remarkable molecule shapes who we are. If you found this video informative and engaging, don't forget to like, share, and subscribe to MedHub for more amazing content about science and the human body.